Thanks very much, everyone, for joining us for today's PNRI Science Matters. This is the last one of the year, but we still have a great schedule coming up next year, so please check out those future talks as well. Today, we're excited to welcome Dr. Eduardo Amorim. Eduardo did his undergraduate, bachelor's, and master's degrees at Universidade de Brasília in Brazil. He stayed in Brazil for his graduate work before coming to the US, where he started with a postdoc in Molly Przeworski's lab at Columbia and made some stops at Stony Brook, UCLA, and in Lausanne, Switzerland as well. In 2021, he started his lab as an assistant professor at Cal State Northridge. Dr. Amorim's group is really interested in using population genetics, anthropology, and ancient DNA to think about evolutionary biology in the context of the human genome. In particular, how can we think about and understand what's happened in the past in the evolutionary history of our genomes to try to understand how our genomes work today in both health and in sickness. Eduardo is very interested in education and outreach. He's on committees focused on diversity, equity, and inclusion for Genetic Society of America, as well as Society for Molecular Biology and Evolution. I think he'll also tell us a little bit about this super cool experiment, shall I say, where he's thought about using the time-tested bit of competition and storytelling and cooperation and community to engage the public in thinking about mammals of all things in the context of science outreach. So we're very excited to have Eduardo, and turn it over to you, Eduardo.
Thank you so much for the introduction, and thank you, Dr. Claudia Carvalho, for the invitation. And of course, thank you, the audience, for being here. I am super excited to talk about two of my passions. I never had the opportunity actually to talk about those two things together. I usually give talks about my research, and I'm really, really excited to share a little bit about my research, but also about the outreach program that we are doing. So the talk is going to be divided in two sections. I'm going to start talking about my research, where I use ancient DNA to reconstruct the evolution of immunological traits in humans. And then the second part, it's going to be focused on my outreach project, or the project that I'm part of, called March Mammal Madness. Yeah, so that's going to be the two divisions. You see here on the right, I use this illustration from the book Through the Looking Glass. And then there is this quote here. Now here you see, it takes all the running you can do to keep in the same place. This is an illustration in a sentence that people associate with the way that we and pathogens co-evolve. So we adapt, the host human organism, we adapt to the presence of pathogens, and then the pathogens adapt to our immunological system as well. And this keeps happening over and over in this complex coevolution evolutionary process. And it's represented by this, so I wanted to bring this. And with this, I want to hook my research. And the way I study this process of host pathogen coevolution, there are many different ways you can study, of course. Most of them are focusing on present-day populations, right? So here I'm representing a human and a pathogen, and they're interacting, and we see here they are towards the end of the timeline. What I'm doing is actually to look in this process using ancient DNA from different time points as well. I can learn a lot about the pathogen with the ancient DNA, but also about the humans. In this talk, I'm going to be focusing on the human part. Eventually, our project will expand to the pathogens as well, using the same type of approach. So basically, to summarize here, the aim of this first part of the talk is going to be to how we're using ancient DNA to reconstruct the evolution of immunological traits in humans. Before talking about the results of the research, I want to talk a little bit about what's ancient DNA, what's some of the characteristics of ancient DNA, and how our field of population genetics have been studying adaptation and natural selection. And so ancient DNA is very similar to most of the analysis of ancient DNA is very similar to DNA that we obtain from fresh tissues like swab or blood. but we are using DNA, we're extracting DNA from human remains or from your species of interest, the remains of that species. Usually we use teeth, and I'll explain why, the advantage of using teeth in a second, but you can also use bones and also material that some humans might have handled in the past and left some DNA, or even soil, for example. And from those, we obtain ancient DNA, which I'm going to be referring here. I'm going to be saying ancient DNA, but sometimes in the slides, you're going to see aDNA, which is the short for ancient DNA. And one thing that a lot of people ask me is like, how you define ancient DNA? I never found a good definition, but I like to define not based on the time, but based on the characteristics and the way we study this ancient DNA, this damaged DNA that I will talk more about in a second. Okay, so this is actually a real tooth that we've been analyzing from, it's 1,000 years old. And the way we do it is that we section it in half. We send half to do isotope analysis to learn about diet and also movement. And the other half, we extract part of the tooth, we pulverize it, and we extract DNA from that part. And that part is what we call the pulp cavity. And the advantage of using tooth and analyzing the pulp cavity, as opposed to other structures like skull or long bones, is that from the tooth, we can actually get DNA from both the human and the pathogens that might be infecting this person in the past. Because the tooth is vascularized, right? It has a blood. So if there is a pathogen in the blood, you can potentially recover from the tooth as well. We have a chance of success when you know that the person is affected because of some like morphological characteristic, for example, for the plague. We have a chance of success of 9%, so it's not very easy to get the pathogens, but we can get sometimes. DNA from humans is easier to get. And using this type of approach, using ancient DNA, people have generated over almost 10,000 human genome sequences already. When I say genome sequences, I mean whole genome sequences that we call like shotgun sequencing, but also SNP capture for genome-wide data. And this data, as of 2021, so 2 years ago, it was distributed this way. And you can see the numbers, how when the field started around 2010, 2014, we have only 43 genomes published during those 5 years. And then in 2020, we have almost 2,000, and this number is growing and growing. Now we have a total, as I see in the top of the slide, we have a total of 10,000 ancient humans that have been sequenced. I'm going to come back to some sort of similar plot like this later, but here you can already see some biases that we have related to the representation of ancient DNA that is mostly focused on Europe and Asia. I'll come back to this later. So using these techniques, using ancient DNA and all this data that we generated, people have answered many questions related to human history and evolution, and people are starting now to answer questions about health as well. And most of you probably know that last year, the Nobel Prize in Physiology or Medicine was given by Svante Pääbo, the professor in Max Planck Institute in Leipzig, for his research concerning the archaic and modern human interactions. So that was truly revolutionary and impacted a lot the field of population genetics and anthropology. And currently, we can use ancient DNA for all these diverse questions. Kinship inference, for example, we can see in an archaeological site who are related to who, which is very important to reconstruct the history of some populations. We have reconstructions of human migration routes. We can recover lost genetic lineages from humans, for example, but also from extinct species. My lab, for example, is studying one type of dog, like a dog, wolf, fox, like a canid from South America that it's extinct, but we got 14 samples from those extinct animals, and we are reconstructing the genome of that animal. So it's pretty cool what you can do with ancient DNA. As I mentioned before, you can also do characterization of ancient human pathogens. And more recently, people started to look into phenotypic reconstruction. And it has been a lot focused on height, but also in like eye colors, hair color, And this triggered some interest in studying natural selection with ancient DNA as well. This figure that I'm showing here, it's a map from a paper from 2016, is an excellent paper describing human adaptations to different environmental pressures. This is all based on not ancient DNA, it's all based on present day populations. And our idea in the ancient DNA field is to do something like this, to be able to understand what are the selective pressures and where they happen in the past. Maybe some signals that disappeared through time. So what are we thinking about when we are using ancient DNA to study that? Because this is a lot of data here, right? So we learned a lot about human adaptations already using present day populations. Why do we want to use ancient DNA, and how could we use it? So that's a good question, like how can we use it? The idea is generally this. So you have time here, a timeline, and then you have a population that it's evolving, and you are able, through ancient DNA sampling, to sample DNA from time 1, time 2, time 3, and time 4, and you see that there is an environmental change between time 2 and time 3. So our idea is, this is not my paper, right? So I'm saying our idea as the field in general. Our idea is to track allele frequency changes that coincide with an environmental change. So here we see that this allele is increasing in frequency from nearly 0 up to nearly 100 between time 2 and time 3, which coincides with the environmental change in the first panel of this figure. So this is the general idea. However, studying ancient DNA comes with challenges and limitations. I brought this meme that I got from a colleague that studies ancient DNA in the Max Planck Institute from on Twitter. I thought it was very funny. It asks, someone is asking two people, and then she asks, are you two friends? And then the two people said no and yes. The person who says no is the population genetics, and the other one says yes is ancient DNA. I think what it means, at least how I interpret, is that we from the ancient DNA field, we think we can use our data to do population genetic inferences, but our data is so difficult, so sometimes low quality, that I think a lot of the methods from population genetics we can actually not apply yet. So there's been a lot of effort from the paleogenomics or the ancient DNA field to develop methods that can actually leverage this information about time series data to learn more about our evolution. And what are the challenges and limitations that I mentioned? Here are some of them. I think all of them that we deal like frequently, they are mostly related to the fact that we are using human remains, right? So those samples are destroyed, are old. So the DNA is degraded. It's fragmented in small pieces, sometimes 30 to 70 base pairs, for example. It has a lot of mutations in CPG sites that are methylated. So you have these transitions that are not corrected because the individual is dead already. So we start to accumulate these mutations in the DNA, and this becomes noise in our inferences. In addition to that, we often find very low DNA content in the material that we have. We sequence a bunch of things, and then maybe 1%, 5% is from the human that you're studying, and all the rest is from like the environment, from the soil and so on. So this is a common problem. We usually have low coverage sequences because DNA is damaged, right, or degraded. And that causes our data to look like there is an excess of homozygotes, because usually we have 1x coverage in our data. In 1x coverage, you only have usually only one allele sequence, right? So it looks like everything is a homozygote. So this is a problem when you're looking at disease inferences or like a natural selection inference, because it looks like everyone is homozygote, and it's actually not the case. There is contamination risk, of course, but we can actually assess that. We can quantify, and we are pretty good in controlling contamination. Then there is another problem, which is biases in sample representation, which is actually not an issue from ancient DNA only, also from genomics, human genetics overall, that there is a strong focus on European populations or white populations. And here, this is a similar figure that I showed before, but this one is from 2016. You see that has a smaller sample size, right? Because in 2016, we had less sequences generated. But you can still see here the overrepresentation of Europeans. But another thing is the underrepresentation of historical samples, specifically in Europe, meaning this blue part of the chart that that is almost not seen here in Europe, right? So in Northern Europe, there is a little bit, but the other one's not. The blue part, when I refer to historical samples, I mean in historical period, but here in this graph is everything that it's more recent than 2,500 years before present. You'll see this BP all the time in the presentation. It means before present. So what we see here is those two things: underrepresentation of Europeans and overrepresentation of Europeans and underrepresentation of historical samples. And I always, I started to work with ancient DNA in 2017, more like deeply with ancient DNA. And for me, it was very curious that no one was studying historical samples, because the historical period in Europe specifically is where many different things happen that are super interesting from the health perspective, but also from the historical and anthropological perspective. One is the several different outbreaks of different diseases. The most well-known is the Yersinia pestis outbreak that we know as Black Death, but there were many others. And there is also the invasion of the Roman Empire by barbarian populations, barbarian people. And this is also related to the origins of many of the European nations. So I was always puzzled why people are not focusing more on this period, that it's so interesting from a biological medical perspective, but also historical and anthropological. So I started to focus on this in 2017, and it culminated in what I'm doing today, which is trying to build this ancient DNA time series data sets, as I refer, to understand history, but also understand evolution in our species. And this work that I'm presenting today, it's funded by a grant from the National Institutes of General Medical Sciences, and it's led by Flavio De Angelis, who is a postdoc in my lab. And he's the person, super creative, super intelligent, who is doing all this work that I'm going to be presenting today. So I wanted to acknowledge him. And I also want to acknowledge Lars Feren-Schmitz. We are using a lot of public data, but the data that we're generating is all in his lab. And he's been an amazing collaborator. I also wanted to highlight him, because those two people are very important for anything that I'm going to talk here. But I also listed the other people that contributed at certain aspects to this project. Some of them are my students, Caroline Solis and David Villarreal. Okay, so this is what we've been generating for Europe. There is also some work we are doing in South America, but I'm not including it here. Just for you to have an idea of what we've been generating new data for, we have from the Mesolithic up to medieval time in different time periods in Europe. Most of the samples are coming from Italy, which is where Flavio comes from. So we have a lot of collaborators there as well, but some coming from Romania and Britain. And this is what we are doing on the side of like generating more data. But today I'm going to be talking about how can we leverage, how can you use this time series genetic data to study natural selection? And this follows the idea that I talked about before, which is trying to match allele frequency changes with environmental changes. And my idea here is that the environmental change would be a disease outbreak. And we have those data, especially in the historical period, because of historical accounts. Sometimes we don't know what's the pathogen, but we can study with ancient DNA. I mean, we don't know what's the pathogen based on the historical accounts, right? But we can get DNA from those samples and see what pathogen it was. And on the other side, there is like this human evolution that I'm showing here, just like changing this shades of gray. So the idea is to do something like this and using the pathogen outbreak, the disease outbreaks as the external environmental change. And as I mentioned before, this is being led by Flavio De Angelis. Now I'm going to start showing a little bit of the data that we have. So this is, so I'm going to start talking about the approach that Flavio developed, which is super smart, super clever. So I showed this figure before about the examples of genes involved in human adaptations. And I didn't mention that before, but those genes, each one of those carry at least one mutation that has a large effect in a phenotype that underlies adaptation to environmental pressure. We know, however, that this type of selection is actually rare in human evolution, which is more common in human evolution. And actually, the different traits that are medically relevant, for example, many of them are polygenic. So looking at those monogenic types of adaptation can teach us a lot, but it doesn't teach you the whole figure. Here I'm showing a figure, a picture from the NIGRI, showing on the left an example of a rare monogenic trait that it's affected by one mutation, and on the right side, a polygenic trait that has several different mutations underlying that trait. But also we call it complex because in addition to being polygenic, it also involves, there is a lot of influence from the environment. And this is very good for what we are studying. So we think studying polygenic adaptation has several advantages over studying monogenic adaptations. First, because most traits of interest are polygenic. I might be wrong when I'm saying this, right? But a lot of the traits that are of interest are polygenic. And we have increased sensitivity to selective pressures because monogenic trait, you need a strong mutation, a large effect mutation to be able to detect, especially with the type of data that we have that it's so poor, right, with ancient DNA. Whereas with polygenic approach, we are able to detect mutations that are even contributing with a small effect to the phenotype that we are studying. And also, it's interesting to study polygenic adaptation because we believe it could be the mode of adaptation that it relates to the dynamic environment with several different environmental pressures happening at the same time and other complexities of human evolution. So we decided to move from the monogenic to the polygenic approach. And we started by thinking about using genome-wide association studies. The acronym is GWAS. And how does a GWAS work? Very briefly here, I believe everyone should be familiar with it, but you sequence genetic variation across the genome for several individuals, thousands of individuals, for example, and then you could split these individuals in two groups. There are other research designs, of course, but you could split into cases and controls, for example, cases carrying a disease and controls are healthy individuals. And then you seek for genetic variants that occur significantly more frequently in one group than in the other. And with that, you are statistically associating a mutation to that trait. And it doesn't necessarily mean that it's a causal, like it's the mutation that is causing the disease, but it is associated with the disease. We leverage this information from GWAS studies, like we usually had hundreds or thousands of variants that are associated with a given trait. And we also get from those the effect that each mutation or each allele has in the trait. So some will have a small effect, some's going to be a little higher, like stronger effect, and so on. And we actually can quantify that. And we can use that information to build this polygenic risk score by adding all the effects of the different variants that we find in an individual, and we can build this polygenic risk score. I'm going to show an example here, but just to explain, the GWAS is done with a population, and then the polygenic risk score can be applied to a different individual or different population. So here is a curve representing people, the different polygenic risk scores, right? And in the y-axis, we have the percentage. We have this type of bell curve. Most people have like an average risk to develop the disease, but some people will be more resistant or low risk. The other one is going to be at a high risk to develop the disease. And here I'm showing the individual like pointed by the arrow. This individual carries two mutations that are risk alleles, for example, and this polygenic risk score is 2, assuming that the effects are the same. And then the other person here by the yellow arrow, it has 7 of those mutations. So the polygenic risk score of this individual is 7. Again, assuming that each mutation contributes with the same effect. So this is what we are doing we are getting ancient DNA, GWAS data, deriving models of polygenic risk score, and applying those polygenic risk scores model to calculate the polygenic risk of some traits of interest using ancient DNA data. And by grouping our samples in specific time periods, we can have this type of distribution, and we can assess if the distribution is changing across time. So that's the whole idea of the framework. The biobanks we are using are the UK Biobank and FinGen. They have about 400,000 individuals of European ancestry in each. And they have about 50 traits that are related to infectious disease-related traits. That is, say, it could be resistant or predisposition to a disease. And from those traits, so 50 traits, we end up by selecting 7 that have significant heritability, and then we can actually do some inferences with that. So those 7 traits are this. The names are a little weird, in my opinion, but this is literally what we get from the description of the trait. If you have more interest, you can check the details on FinGen and UK Biobank. But basically, one thing that I want to show here, highlight here, is that there are most of the traits here are very general, right? Like intestinal infections, diarrhea, and gastrointestinal infections, all the bacterial diseases. So I'm going to come back to this later also, but I want to highlight this, that it's those traits are very general, are not very specific, except this erysipelas, that it's more specific. Okay, so once we have that data, we have those traits that we are interested, like has high heritability. We want to get the polygenic risk score model to calculate on our for our samples, as I mentioned before. So the samples we are using, the ancient DNA samples, are publicly available ancient DNA data in this AADR database, and includes data sets from several ancient DNA studies. All the ones that are publicly available, they aggregated into this database. The region we are focusing is Western Eurasia, and we are using pseudohaploid data, which is a way that we control for problems that we have with ancient DNA data, that it's usually high frequency of homozygotes. So what we do is that we transform everyone in a homozygote. So the sequences we use is like pseudohaploid. So we introduce the same noise across all the samples. The genetic variants we're using is what we call the 1240K SNPs. So it's basically 1.1 million autosomal SNPs that are spread all over the genome, and we get the samples that have at least 600,000 SNPs or SNVs, and that results in us having 2,400 individuals in our data set. And those we split them in groups of whatever number of individuals in each bin, but the bins are either 200 or 400 years interval. So we group them by age, and we get them like based on that that's the way we group them, by either 200 or 400 years intervals. And our time transects go from 200 years before present up to 10,000 years before present. So this is the general approach. Again, just to show the idea here. And let's go to the results. So the idea here, as I mentioned before, is I have a PRS distribution before the outbreak, then I have the outbreak. And the question here is this PRS distribution changing after the outbreak? Can we detect some changes? It could go up or down, or even could be like over dispersed. And we do find those 7 traits, 4 of them are actually associated with what we call the proxy phenotype, which is this time transects. So we see that for 4 of them, we see that are significant changes in the distribution of PRS across the different time points. And so those are the 4 traits. There are the Q value there. It reflects the association between the time variable or time transects with the PRS. After multiple test correction, we also use some covariates like sex, geography, like the position, latitude, and longitude, the 21st principal components that summarize genetic ancestry and population structure. And we also control for correlation with other traits, because those traits are, they share some genetic bases. So we also have to be careful with that. This is what we see for the thin gen traits. There are 2 there and 2 for the UK Biobank. And we can see here, each of these violins are a time transect of 400 years, and it goes from the present in the left to the past on the right. And we can see here that there is a shift in the median of this PRS distribution, right? That we can actually visually see, but it's also statistically significant. And you can also see some sort of over dispersion, especially on the left side of the plot on the upper one. And we do not see this visually here, but statistically, we also detect those changes here in the UK Biobank. So our next question was, what are the time transects that are actually contributing to these differences that we detect with the statistical test? So what we did is to compare these distributions pairwise, so 2 time transects per time. And what we see is that those 3 time transects, so from 600 years before present up to 1800 years before present, all this, we see those 3 time transects being always showing differences between them and other time transects in relation to the PRS distribution. And curiously, those 3 coincide with major epidemics from the past. One is the Antonine plague, the other one is the Justinian plague, and then the other one is the Black Death. And here's some characteristics of them. The Antonine plague is the oldest one, so it was 1800 years. Yeah, it's the oldest one in this. The time period is there, and it's likely caused by either smallpox or measles. And the 2 other ones, the Justinian and the Black Death, were caused by the Yersinia pestis. And everyone probably heard about the Black Death, right? It's the most fatal pandemic recorded in human history. It's estimated that at least 25% of the population of Western Eurasia died in that period. So it's logical to think about those things, specifically the Black Death. Here we have better numbers, as a very strong selective pressure. And that's actually what we see happening here. We see that potentially those pandemics in the past are affecting the distribution of polygenic risk score of immunological traits. The other question that we have, and this is towards the end of this first part of the presentation, is what other traits are being affected by this? So there was a paper that actually answers that. They also used ancient DNA, and they used polygenic risk scores to study infectious disease risk. And they see that whenever you have an increase in resistance, you also have an increase in inflammatory disease risk. So the infectious disease, you have resistance against it, and it grows together with your risk for increased inflammatory disease. This is really cool work, and it shows this complex way that our immune system adapts to selective pressures, right? And what we did is it's less complicated than what they did. It's just basically getting the genes that were showing shifts in allele frequencies and getting the name of those genes and running them through like a gene ontology tool to see which biological pathways were involved with those genes. And by doing so, we believe we are inferring additional pathways that could potentially be impacted by this adaptation to the pathogens that we described. And you can see here, there is a bunch of things related to pathogens, like clearly related to immunological system, but there is a lot of that are actually not directly related with the immunological system, but are indirectly, like lipoprotein particle clearance, pigment granule localization, insulin response, and so on. So just to summarize this first part, we show that the evolutionary significance of host pathogen interactions, right? We show that pathogens have been a crucial selective pressure, had a crucial role in shaping our genomes over time. We also showed that using ancient DNA, we can gain additional insights about evolutionary aspects and also health related aspects of our biology. Specifically, we know sometimes what's the date of some event happened, but also how strong that event was in our genome, or like the selective pressure, by seeing how much of this allele frequencies changes, how fast they change. Third, I put here that despite the limitations in ancient DNA, we do observe with this approach that we are developing changes in the polygenic profile during very important events in our history. And last, our results provide insights into the systemic involvement of the human host genome in the dynamic host pathogen relationship, meaning, as you see, the traits that we detect changes related to time are very broad. They're not specific to one pathogen or another, but also we see using the gene ontology approach that not only the immunological system, but other systems are also being impacted. We are still working on this paper in this manuscript. We are hoping to submit early next year. So if you have any comments, I would love to hear them. And now I'm going to change to the science outreach project, which is my other passion. And should I stop now or continue? And then we have questions for the research part as well, together.
Just go ahead through the end if that's okay with you.
Yeah, perfect. So I think I'm gonna have like 12 minutes to talk about this. So I'll it's the first time I'm talking about the project for like in a seminar like this. So I hope it fits in the correct time. But so I'm gonna be talking about the science outreach project called March Mammal Madness. And what is March Mammal Madness? I'm gonna be abbreviating it as MMM. So it's a science outreach project in the form of a simulated tournament between 65 animals implemented every year via Twitter that runs over the course of several weeks in March, involving hundreds of thousands of students nationwide. This year we had 600,000, so more than 500,000 students involved, like playing the game. So if you want to know more about the project, you can scan the QR code from the eLife paper. The other one in the bottom is unfortunately not freely accessible, but you can reach out to me if you're interested and I can send it to you. So how does MMM work? I'm going to be talking about the player's perspective first, and then I'm going to tell you the developers, I would say, perspective afterwards. So you will see how things happen. But let's start with the player's perspective. So each, in late February, we released these brackets like this. This is the one from this year. And it has 65 animals. You will see that each division, we have 4 divisions: Mighty Stripes, Itty Bitty Convexity, Animal Engineers, and Dead Bods. Each one has 16 animals, and one of them is here, the bottom right, has 17, because we have this wild card winner, which is the first battle. That's how the tournament starts. And you will see here, there are like funny names. The divisions have always like weird or funny names. And then the students receive this, and they pick their favorite combatant, or like the ones that they think it's going to win. You see some numbers here besides the name of the animal. Let me see if you can see, like there is okapi in number 1, and then 4 striped grass mouse in number 16. So that's what we call the seed. It's proportional to the size of not proportional, but it's the rank of the size of the animal. So okapi is the largest animal in that division, and the 4 striped grass mouse is the smallest animal. So it's seed 16. Okay, so and then this is some text that we use in our tweets, or in our material. But here I'm showing some of them, and I would like you to pick one. I'm just gonna go over them like kind of quickly, but maybe pick which animal do you think is gonna win? So here, for example, okapi is the largest one, but maybe it's not the most aggressive, right? Maybe it's the striped hyena, which is the 3rd largest animal here, because hyenas are aggressive, right? I don't know. So think about this. I'm going to pass through this, and you pick what's your favorite, like what animal do you think it would win if those animals were fighting pair by pair, right? So here there is the sea otter, that is the largest one. Those are small animals. We were bringing small animals that lost in previous tournaments, and we brought them back. So that's why we call itty bitty convexity. Usually those animals don't win, spoiler. So I'm going to move to the next one, which is the animal engineers. So you will see here also actually that two of the divisions are exclusively mammals, and the other two are not exclusively animals. So they can include anything like the Brazilian bird, I forgot the name in English, Rufus ornero. So this bird that builds this nest that it's like a cave, and also this fish that builds this thing in the bottom of the ocean. And then all the animals like Homo habilis, for example, and all the animals that build something. So maybe you see something here that you like maybe the golden eagle, I don't know, that you think it's going to win. So let's move to the last division, which is this dead bogs. I'm going to read here because this name is funny, right? So I'm going to read here just to explain a little bit. So although March Mammal Madness routinely consider animal traits for combat, for some species, males play really important roles in nurturing youngs. So usually our divisions are based on some sort of adaptation that the animal has. But this time we focused on species that the male is the one that take cares of the youngs, or actually like take care of the young, or like let's say here, combator has adaptations for behavioral care, some with anatomy for protecting young, and some can nourish the young. So this is the last thing that I'm showing. Maybe you want to have a quick look here to pick your favorite. Can I see which one? Can you put in the chat, maybe a few people at least, which animal do you think is going to win in this combat? Maybe I'll give like a minute. Zebra. Anyone else? Badger, bat. I'm going to say which one I picked. It was the golden eagle, because I thought, so I have access to the outcomes, but I don't want to look at the outcomes before I pick my winner. So I picked the golden eagle. I'm going to just write here, golden eagle. And because never non-mammal won, so I thought maybe it's this year, it's a golden eagle. Come on. Okay, so we have a few animals here. Let's see if anyone got vein octopus, side star jackal. Okay, very good guesses, but no one got it. Not it's actually okay. So which one? So the one that won was this. It was the wolverine. So just a quick pause here. We built this like Wheaties. I'm not American, as you noticed, so I don't know what it means, but I was told that this like cereal box, they usually feature like winners of some sort of sport. So we make this fake Wheaties boxes to for the champion. And this year was the Wolverine. And here's an example of a battle. I'm gonna skip for the interest of time, but basically what we do is that we narrate this in, this is some examples of the narration. I'm going to skip this to the next part. So we describe the environment that it's happening. It's usually like a real place that exists. It's always a real place that exists. And then there's so many different things that happen. In this case, there was the Wolverine. Oh, let me see if the Wolverine is there in this environment, the Sierras. And then we use the magic to transfer the other animal to that place. That's the only fake thing that we do. Everything else is based on science. But we transfer the animals to one animal to the other place using magic. And once we do that, there is a bunch of things that can happen. The narrators that craft this story. In this case, Katie is the creator of this program. She's writing this final battle, and then there is this bear cub that comes, and then there is the mom that comes, and then the two animals that are the focus here is the okapi and the wolverine, right? They run away from the bear, and they end up in the river, and then they have some fight there. They hurt each other, and then eventually the okapi runs away. And then that means that you're abandoning the field of battle, right? So you are losing, which is what we call technical KO. I'm going to come back to this later for to see like what is the type of ends, like how animals mean in a second. So some things to note. These are live tweet narrations. It has a lot of pauses for the dramatic effect. So that makes the story more engaging, almost like a real sport match. So it looks like you are watching that on TV, you know. I usually know, I work with the genetics part, and I will talk about it later, but so I don't do the narrations myself, so I actually see the narrations happening, and I actually, my heart sometimes go like, because it is, the narrators are so good in creating this story that a lot of people are engaged. So with the exception of MM Magic, we use to transport the animal to the battlefield. Everything else that happens is based on scientific literature, and or in images of naturalists, like some sort of behavior that we see from pictures that some animals did. We use that to illustrate the stories. There is a lot of engagement from players, like people retweeting, people quote tweeting, and there is a fan base. It's really cool. They create a lot of different things, and it's very funny. So one thing that I want to say, no one picked Wolverine, right? So, but our motto is, if you're learning, you're winning. So now I want to ask you this, like, how could you make a better informed decision? For the interest of time, I'm going to give you the answer. So you would do research about these animals, right? So players are given about 10 days to make bracket predictions. Most players are high school. I think I put it wrong. It's not high school. It's like students from schools, young students. And the school educators are teaching life sciences, and they can request MMM materials in February, earlier than 10 days before the tournament, to come up with lesson plans for the classes. And here in the bottom, we see that 75% of the teachers, they use MMM within the curriculum, and with the aim to strengthen, extend, and refine student understanding of course themes and topics. So what I want to tell you here is that although you didn't pick the correct animal that won, you could have picked it if you had studied more and read about these animals. So that's what we want the students to do. And that's the way we can engage students, because they are playing a game, so they are very excited with it. From the creator's perspective, I'm going to go fast here. We are 60 people here, some of us. We have geneticists, artists, narrators, librarians, educators, etc. This is our illustrators, some of the illustrations that they do for the tournament. Who makes the decisions? Like who tells who's going to win? Katie chooses the 4 divisions and the arenas, the ecosystems that the battles will happen. And she picks most of the combatants, but all of us suggest, like a few months in advance, who we would like to be featured. This is a list of all the different taxa that have been included. This is the types of environments. Each year, there's always 4. So after she picks that, and we have a list of 65 animals, we give seed numbers for each combatant based on their weight. So 1 is the heaviest, and then 16 is the lowest, the lightest. And then within each division, animals are randomly paired. And based on the seed, you have a larger advantage if you have a higher, closer to 1, right? You have a higher chance of winning. And then also there is a home court advantage or disadvantage if you're in an environment that is not the one that you adapted or any more adapted. And then we use a random number generator to decide who's going to win in each match. So based on that, we have the outcomes, and the narrators have to come up with a story based on scientific literature to describe that outcome. And here are the different types of battle outcomes. Usually we have this technical KO where there is like some animal is hurting each other and the other one runs away, because we are trying to do everything most scientifically possible if those 2 animals actually meet in the field. So most animals will actually run away. So withdrawal and technical KO is the most common way of the outcome of the battles. So this I mentioned before, we are all based in literature. I'm showing here the numbers of papers that we use each year. So we are actually getting papers from the literature and advertising that through so many people, right? So we are doing this type of science outreach as well. The battles, okay, last thing here, the narrator uses those 4 approaches to science outreach. For me, the most important things is the gamification and the live aspect of the game using social media. And reach and impact, here's some numbers. We have this year 6,000 teachers requesting materials. And we estimate that 600,000 people played March Mammal Madness this year. This is the distribution. We have very lower numbers, let's say in Mountain West and Midwest Plains. The rest of the country is actually where most of the players come from. We have players coming from urban, suburban, and rural areas, so it's pretty diverse. This is some qualitative assessment. I'm gonna skip, but basically, why the teachers like it so much? Because the students get very excited. It sparks interest in science, even from students that never like to do science or study science. And yeah, so this is some of the feedback that we have. We have a lot of those. I really love to read them. And this is the types of things that we see that the teachers send to us, or the players. You see like memes and walls with drawings and etc. Like winners they get in their school, for example, like a little trophy and so on. So this is really engaging. A lot of people participate. Is really cool. And that's it. So I'm gonna skip this. If you wanna have a look at this, you can do a screenshot and get later. And then I'm gonna thank everyone here, all my collaborators. Of course, Dr. Claudia Carvalho for inviting me, and everyone from the Pacific Northwest Research Institute that I talked with, and everyone who came here to this talk. I had an amazing meeting with 3 of the PIs before, and also, of course, Flavio De Angelis for doing the hard work that I talked about today. And with that, we have 7 minutes for questions. I can stay a little longer if you can.
That's great. Thank you, Eduardo. That's super cool. Is it always like a number 1 or 2 seed that wins the bracket, or have you had some upsets of the lower seeded animals coming through as winners?
So because of the random number generator, right? So we do have upsets, very common. So this, the Wolverine is seed 3, for example. So it is a little bit of luck. Maybe the environment was favorable for one animal, not the other. But there is at least 1 or 2 upsets that we receive kind of like rude messages from students because they get really upset that, like, let's say a lion lost to a rabbit for whatever reason. And then it's impossible, right? But sometimes it happens because it's a random number generator. Of course, the probability is small, but then it's very funny. Katie is the one who handles that. So she has a lot of like, she knows how to handle it, but she shares with us the comments, and it's very funny because people get really passionate. It's like sports, really. Like if your team lose, you get really angry.
We need to sneak a virus into there, I think. Maybe it will dominate. But
we had so the story is that Katie's an anthropologist, and she studies milk, like feeding, breastfeeding, and she studies all the mammals, not just humans. And she came up with this idea. She's super creative. And then there was one year that she was in sabbatical. I think was the 3rd or 4th year. So someone created a category, because the second most important person there is this other guy who is not a mammologist. He studies plants, I think. And then he made a division called When the Cat is Away. Cat with K because of Katie. So when the cat is away, let's put animals that are not mammals. So we started to include mammals then. And then there was a plant at certain point. There was a hydra last year. There are like some different animals. There was also like some dogs. Anyway, sometimes we there are some different animals, but I don't think there was ever a virus. But it's a cool thing to think. Oh, actually, we had a category called Why Not Both? And that category was animals that have either an adaptation that, like a hairy frog, for example, like a frog that grows something in the skin that looks like hair. So it's like, is it hair or skin? So why not both? And then there was also some animals that are a symbiont. And then I remember that was one of my suggestions. There was a salamander that has an algae that is a symbiont. So we had some organisms that are like unicellular, but in that context, at least this one I remember.
Very cool. Questions for Eduardo? Please feel free to unmute and ask.
Oh, someone said I participated in March Mammal Madness in the past. Oh, nice, cool. I love to play it, and we see it looks like it's for kids, right? But there is a lot of adults that play. There is also families that play with their kids. So they post photos online, like Twitter, everyone, like, because it's late night. So like the kids with pajamas, the parents, and then like they take a photo and send. At the university when there was proctoring an exam, and then this guy came to talk to the proctor. I was giving an exam, and the proctor told me, like, as a secret, like, to not disturb the exam, right? He said, I play March Mammal Madness. So it's like there is adults playing, and I think it's really cool. Yeah. It's so powerful, I think. I'm so proud to be part of it. It's so powerful that everyone is engaged, everyone who sees it gets involved.
I had a question about the PRS shifts and the GWAS that you did there. Did you look for overlaps in those GWAS peaks with like lupus GWAS hits or other sort of autoimmune GWAS studies that have been done?
We didn't, but let me find, there was this paper who did exactly this. They were, yeah, so this paper, if you want to screenshot, because they actually look into that, but we haven't done that. I would like to do that, but I think we're going to move on without this point to get the manuscript faster out. But that would be like something I would like to do.
Eduardo, I found it fascinating that you are doing PRS in ancient DNA. So I like you put a lot of the limitations, but I was wondering because you don't have so many choices, right? For the DNA, although you have a lot of there seems to be, I didn't know there was like a database of ancient DNA sequencing DNA, I'm assuming, or is this SNP array? What
Oh,
type of data?
most of the data is SNP array,
Oh, SNP
but
array, okay.
they have both there. But we usually work with the SNP array because I don't know how much, I would say like maybe 2/3 is generated with SNP array. So we just get the subset of the data when we have the whole genome to the SNP array SNPs. Yeah, go ahead, sorry.
Yeah, no, I was just curious, how do you do to control what the comparisons you are doing, because of the limitation in terms of material? And also I'm wondering because PRS is very prone, like diversity is an important factor for the PRS, right? So how do you control for that?
I think so. One thing is that we use the PCs as a covariate. So we know, because some allele frequencies can change because of migration, right? So a population comes bringing the new allele and then increasing frequency. I mean, I think it's natural selection, but it's not. So we control for that with the PCs. I believe it's a good way to do it. If anyone has an idea, let me know. But the other thing is this portability issue. I think you were also referring to this, I believe. Like I didn't show this slide here, but we are not sure, as far as I know, we are still unsure if the GWAS results on present day can be portable, it can be translated into like with ancient individuals. We don't know that. The reason we use European-based GWAS is because most of our data is from European ancient DNA samples. So we believe we are doing better than using other data, right? So it's the best we can do is this. But here we see, for example, the relative predictive performance with UK polygenic score translated to other populations. So we see that for UK, it's really good. And then as you go to other populations in Europe, it's a little worse. And then when you move out to another continent, it's much worse. So there are issues here. we don't know how to correct for that.
Mhm.
What some people have been doing is to compare the polygenic risk score for height and the actual height of the individual that you can actually measure. And we can see that in moments of bad health, like in the Neolithic, for example, when the agriculture comes to Europe, you start to have a lot of like poor alimentation, but also like a lot of disease because of the larger societies, the living with animals, like close to animals. So you start to see impact in health. And what we see is that the polygenic risk scores for height don't match in that period, but match in other periods. So height is subject to health, like how the individual grows, right? It's not just the polygenic risk score. So there is that, but the positive side of that is that for other moments where health was less of an issue, we see it's concordant, the polygenic risk score with the height of the person. So it's that's the only thing that I know that we can actually compare and measure if the polygenic score is correct. Other than that, we're just assuming those people were prone to infections or not. But that's a big caveat. And in the manuscript, we discuss that as an issue.
Okay, thank you.
Thank you.
I was wondering if I could have a quick follow-up on that. That's really interesting how you're doing the principal components analysis to try to get at that. Would you be able to use the principal components, validate that method on the data that you just showed, looking at the people from different locations, and if you add the principal components analysis on the modern data from people of different locations, would that correct it, and that might be able to sort of validate that practice based on modern results?
Never thought of that. I will actually write down your name and this idea. Yeah, no, I never thought about that, because if what I'm doing is correct, I could potentially correct that relative performance, right, of the like translating. Actually, so Flavio understands more of this than I. Last year, I think last year at the ASHG in LA, in the meeting, there was a lot of talks about portability of GWAS. So there is a lot of research on that. I'm not as updated as I should, but I will think about this. As a really awesome, like interesting suggestion, and I'll talk to Flavio about it. Thank you.
Cool. Thanks. It's really interesting talk.
Thanks.
Any other questions for Dr. Amorim? All right, well, thank you very much again for taking the time to present this awesome data to us. And please join us in January for a seminar from Gaida Mirza from Seattle Children's. And again, a big thank you to Eduardo for joining us.
Thank you so much.